I'm Lucy Ireland, and I'm a PhD student in Dr. Naomi Elgo's research team at the University of Liverpool. Our research team investigates the interactions which exist between solid tumours and their surrounding non-malignant cells, and how these interactions may be promoting tumour progression. One of the cancers we are currently investigating is pancreatic cancer. Pancreatic cancer is one of the leading causes of cancer death, and the therapies currently available are not very effective. This graph from Cancer Research UK shows trends in cancer survival from 1971 to 2011. Pancreatic cancer is in purple, and unfortunately down at the bottom, you can see there's only been a 1% increase in overall survival from 2% to 3% over 40 years. This is a very dismal prognosis. This schematic shows a simplified solid tumour. Tumour-associated macrophages, known as TAMs, and fibroblasts are non-cancerous cells which are found within solid tumours. This includes pancreatic cancer. There has been accumulating evidence that these cells can promote tumour progression, increase resistance to chemotherapy, and also aid the metastasis of tumour cells from their primary tumour site to a secondary tumour site within the body. However, the exact mechanism by which these cells contribute to pancreatic cancer progression and their resistance to chemotherapy is not entirely understood. However, we have recently discovered that macrophages and fibroblasts can directly support the chemotherapy resistance of pancreatic cancer cells through the action of a secreted protein. The secreted protein is released by the macrophages and the fibroblasts, and then binds to the cancer cells and activates a survival signalling pathway, therefore making the pancreatic cancer cells resistant to chemotherapy. We've been able to demonstrate in our preclinical models that a combination treatment of the standard chemotherapy and an inhibitor of this secreted protein can prevent the survival signalling pathway from being activated and making the pancreatic cancer cells more susceptible to chemotherapy, reducing tumour growth. To confirm these results in the human pancreatic cancer patients, we then looked at tissue biopsies from pancreatic cancer patients and stained them for markers of the survival signalling pathway, which is activated by the secreted protein. This is shown here in red and brown staining. When we quantified all of these tissue biopsy staining, we found that 72% of patients had the activated survival signalling. This means that 72% of patients may be able to benefit from a combination treatment of the standard chemotherapy and an inhibitor of the secreted protein. These results are really exciting because not only have they uncovered a mechanism by which pancreatic cancer cells become chemo-resistant, but it's also described a combination treatment which may be more effective in treating this lethal disease. We'd like to thank our sponsors and our supporters, and if you'd like to see any more of our research, please visit our lab website, www.theelgolab.org. Thank you.